In this video, I'm going to discuss the mechanism of pathogenesis of the chronic granulomatous disease or CGD. But in order to understand the mechanism of pathogenesis of this disorder, I will first have to explain the mechanism through which neutrophils will destroy pathogens. So, via the respiratory burst process, oxygen will be converted into the superoxide radical via the NADPH Oxidase enzyme. And then later on, superoxide radical will be converted into the hydrogen peroxide by the superoxide dismutase, and now this H2O2 will be combined with the chloride by the myeloperoxidase enzyme to yield hypochlorous acid. And so once a bacteria is phagocytized by the neutrophils, this hypochlorous acid will be used to destroy the bacteria. Now, patients that have CGD, they lack the enzyme NADPH oxidase, so therefore they wouldn't be able to produce the superoxide radical from the oxygen. However, the good news is that the hydrogen peroxide can still be produced as a metabolite of bacteria, and thus this hydrogen peroxide that is produced by the bacteria can be combined by the chloride via the myeloperoxidase enzyme to give hypochlorous acid and thus destroy these bacteria. Now, the problem arises when these patients with CGD are infected with bacteria that are catalase positive. And catalase is the enzyme that will break down H2O2 or hydrogen peroxide into water and oxygen. So by destroying the hydrogen peroxides, therefore there won't be any more H2O2 to combine with chloride, and thus these patients would be susceptible to infection by catalase positive organisms. Now, before I'm moving on to CGD and explaining that in more details, I would like to let you know that the superoxide radical and hydrogen peroxide, these are toxic compounds that can destroy the lipid membranes, proteins, as well as DNA. So therefore, these are referred to as reactive oxygen species. And so therefore, the body has defense mechanism in order to neutralize these compounds. And these include the enzymes superoxide dismutase, catalase, as well as the glutathione peroxidase. So these are antioxidant enzymes. And the glutathione peroxidase is dependent on the reduced glutathione in order to detoxify hydrogen peroxide. So here, as you can see in this reaction, glutathione peroxidase will neutralize H2O2 by combining it with the reduced form of glutathione to yield water and the oxidized form of GSSG. And here we can add half of O2 as well. And then the oxidized form of glutathione can now be converted back into the reduced form via the NADPH and the glutathione reductase enzyme. So just to summarize the findings, these superoxide dismutase, catalase, and glutathione peroxidase are the antioxidant enzymes that protect the cell components from the reactive oxygen species. Alright, so next we can go over the mechanism of pathogenesis of the chronic granulomatous disease or CGD. So as we just discussed, defects in the NADPH oxidase by the phagocytes, including the neutrophils, will result in the CGD. And these patients present with recurrent bacterial and fungal infections as well as the granuloma formation, for which the name Granulomatous has been given to this disorder. Now, infections are generally caused by catalase-positive organisms. As I just discussed, the bacteria will produce hydrogen peroxides as part of their metabolite products, and thus the CGD patients can still use H2O2 in order to produce the hypochlorous acid and destroy those bacteria. However, those microorganisms that are catalase positive will break down H2O2 into water and oxygen, and thus since there is no more H2O2 left in CGD patients, therefore they cannot destroy these microorganisms. And catalase positive microorganisms include the space organisms, which include Staphylococcus aureus, Pseudomonas, Aspergillus, Candida, as well as E. coli. And then nocardia is also another common cause of infection in patients with CGD. And then one method for diagnosing CGD is to check for the function of neutrophils in the production of the superoxide radical. So for that, you can use the NBT or nitroblue tetrazolium test. And so what happens with the NBT is that the superoxide radical will convert NBT into the blue formazan, which forms precipitate inside the cells. So if we have a cell and we do the NBT testing in these cells, what happens is that if there is a normal production of the superoxide radical by the NADPH oxidase enzyme, then they would turn into the dark blue precipitates of the formazan. So this is a normal test with the formazan that is being produced. However, in patients that have CGD, there won't be any superoxide radical to be formed, and thus there won't be any change in the color of the NBT. So NBT is normally a yellow color, and so it stays yellow. And this is an abnormal test where there is no formazan precipitates that are being formed. And that concludes discussion of the CGD and the mechanism through which respiratory burst contributes to the destruction of the pathogens.